Welcome to the second quarterly lecture series of the Network for Ocean Worlds. NOW is a NASA astrobiology program research coordination network established to advance comparative studies to characterize Earth and the other ocean worlds across their interiors, oceans, and cryospheres, to investigate their habitability, to search for biosignatures, and to understand life in relevant ocean world analogs and beyond. Our first season for the quarterly lecture series is focused on life on ocean worlds. The first episode was an introduction to the cryospheres of the ocean worlds and Earth, and today we'll be hearing about life on the seafloors and in the oceans of Earth and the oceans of the outer solar system. Today's talks will be given by Dr. Julie Huber from the Woods Hole Oceanographic Institution and by Dr. Kevin Hand from the NASA Jet Propulsion Laboratory. Thank you for joining us today.
Good afternoon. I'm happy to be here today. I, of course, have to admit that thinking about life on ocean worlds has not been a huge part of my life the last few months. This is my third time in my office in that time, but I came in because I'm also homeschooling 2 children, and an hour and a half without interruption is pretty rare. However, the oceans have always been a great escape for me, both personally and professionally, so I'm looking forward to escaping to talking about life on the seafloor and in the oceans, both here and on Earth, with Kevin. So everything I'm going to be talking about today here on Earth, of course, is couched within thinking about oceans beyond Earth. This, of course, is an image of a number of planetary bodies in our solar system that we believe have oceans that Kevin's going to be talking about. And now that we know Earth is not unique in having an ocean, we can think about this very profound question: does the biology that we know about here in Earth's oceans work beyond Earth? So the life forms I'm going to be focusing on today are microbial life, microbes, and this is a universal tree of life showing all the DNA-based known life on our planet. There are a few different domains of life here, but the main 2 that are microbial are shown here in the blue and brown, the bacteria and the archaea. You are located way out there in the green in the eukaryotic branch of life. So the rest of this tree is microbial, meaning single-celled organisms. The reason there are so many branches in that part of the tree is because microbes have had billions of years to evolve and diversify and fill out almost every known niche on our planet. So Earth is about 4.5 billion years old, and life was thought to have originated between about 3.5 and 3.8 billion years ago. So life, microbial life, was the main mode of operation for most of Earth's history. So I did want to mention that in addition to microbes, we're beginning to think a lot more about viruses, not only in the ocean, but also in an astrobiological context. So you'll notice that I didn't have viruses on my tree of life, and that's because they don't fit on that tree. They can be made up of DNA or RNA, much like other forms of life, but instead, many people do not even consider viruses living organisms, but instead an infectious agent. And that's because they can only replicate inside an organism, a host cell. So all types of organisms, plants, microbes, animals, are susceptible to specific viruses. And as oceanographers, we're learning that viruses are more and more important in the ocean. In fact, if you lined up all of the viruses end to end from the ocean, they would stretch across the Milky Way about 100 times. I've also thrown in here the least popular virus in the world right now, the coronavirus. Again, this is an RNA-based virus. It's named after these sugary proteins, the corona that surrounds it. And I just wanted to mention that there are no known coronaviruses in the ocean. But by studying microbes and viruses in the ocean, especially in the deep sea, we've learned a lot about how they survive and the core requirements for life for these microbes to survive on Earth. The first requirement, we believe, is they need liquid water. The second is that they meet a variety of physiochemical parameters, including things like temperature, and in this case, the upper known temperature limit of life is 122 degrees Celsius, and as I'll be talking about in great length today, a chemical disequilibrium that they can exploit for growth, and I'm going to get into that much more today. So I study both microbes and viruses in the deepest part of the ocean, and this is my favorite map of the ocean, which is a little bit strange because the water has actually been subtracted out of this image. But the reason I like it so much is because you can see all of the amazing features on the seafloor, and that is due to the process of plate tectonics. And so you can see the world's longest mountain range, the mid-Atlantic, the mid-ocean ridge running through the Atlantic Ocean into the Indian and around the Pacific. And we have plates spreading apart, plates slamming into one another, and we have new ocean crust constantly being formed at these mid-ocean ridges and other places on the seafloor. About 75% of the volcanism of our planet actually occurs underwater, and it really is true what you've heard, that we have higher resolution maps of Mars, for example, than we do of the bottom of the ocean. And that's because making a map through all of this seawater is very hard. Now, I'm going to be focusing mainly today on places on the seafloor where we have these volcanic features or hydrothermal systems. Now, these are formed when seawater reacts with a heat source beneath the crust, and the seawater is chemically modified, producing a variety of features on the seafloor. And the one I'm showing here is hopefully familiar to most of you. These are black smokers, high-temperature hydrothermal systems. These are where hydrothermal vents were first discovered, this type of system, in 1977. Sorry, somebody's asking me something here. I don't know what that was. And they were first discovered in 1977 along the Galapagos spreading ridge. And these high-temperature black smokers are basically this rich chemical-laden fluid coming out onto the seafloor and the minerals precipitating out. Over there on the right, at the lower temperature end spectrum, you'll see tube worms. Now, when vents were first discovered, nobody thought life could exist in the deep sea. They thought all life had to rely on photosynthetic energy. But in fact, those tube worm guts are stuffed full of microbes, and those microbes are eating hydrogen sulfide and oxygen to produce carbon for their tube worm host. So the fact that life can not only survive and thrive in this environment has to do with this very fundamental reaction that's been taking place for billions of years on our planet, and that is when seawater moves through oceanic crusts, a series of chemical reactions takes place and releases different energy sources. And in the imagery I just showed you, the water-rock reaction taking place is when basaltic rock is reacting with water and it creates things like hydrogen sulfide and iron. And again, this is perhaps the most well-known type of hydrothermal system, hopefully showing in a video for you right now. These are the world's deepest hydrothermal chimneys at about 5 kilometers deep. And again, you can just see this gushing black fluid that is rich in iron sulfide and the very diverse animal life that these systems can host. However, for the first 15, maybe 20 years of hydrothermal vent research, this is what we knew about. But in fact, we now know that there are other water rock reactions that can create energy sources and support diverse communities in the deep sea. And so in this schematic here, I'm showing when those lower crustal rocks, in this case peridotites, react with seawater, you get very different energy sources. You get things like hydrogen, methane, and simple organic molecules like formate, ethane, and propane. Those systems in turn look very different. And if there's a video showing for you right now, this is the Von Dam hydrothermal vent field, which is only located about 20 kilometers from the first video I showed you. But this is an Ultramafic host system that we found in 2010, a large group of us led here out of Howell. And it has a really unusual chemistry, which I'll show a little bit more about in just a moment. But importantly, when we found this site, we hadn't predicted a place like it would exist. And we realized when the Cassini data came back a few years later that this might actually be a really good analog site for Saturn's moon, Enceladus. And I'm pointing this out because I think, especially in the astrobiological community, we often fall into this trap of the best analog site. And I just want to emphasize that since ultramafic habitats were discovered by scientists, we continue to find many other types. And shown up here are 5 ultramafic habitats, all in the marine realm, from very shallow over there in Perny Bay, that's a scuba diver, so I think that's about a 10-meter deep site, to the Lost City site, which is at about 700 meters, to mud volcanoes, the South Chamorro site, and those also associated with mid-ocean ridges. And these ultramafic sites, even though they have similar fundamental water-rock reactions taking place, they're very different geochemically. And here is just a little bit of data showing, for example, that they range in temperature from very cold to very hot, pH from extremely acidic to extremely basic, and what they often share in common is extremely high concentrations of hydrogen and methane. So I just wanted to emphasize that we don't want to get trapped by what we already know about here on Earth, because as oceanographers, we are continuously making new discoveries about how these water rock reactions can work on our planet. And just to highlight that, here are a few more weird places I visited on the seafloor. This is footage from the Ocean Exploration Trust in a partnership with our subsea NASA-funded project of the Loihi Seamount. Now, this is a mid-plate seamount in the middle of the Pacific Plate near the coast of Hawaii, and the volcano here is just covered in iron. So you don't see any of those lush animal communities, and it looks very different because the geochemical energy supplying this is also quite different. And finally, this is yet another dramatically different site shown in the western Pacific Ocean. This is the West Mata volcano. This is about a mile beneath the sea, and this is along an arc where you have a huge amount of remelt and volcanism. When we witnessed this eruption 10 years ago, 12 years ago? Maybe it was 12 years ago. That was actually the first time any deep sea eruption had been seen in person through the eyes of a remotely operated vehicle. So if you walk away with nothing else today, I want you to walk away knowing that there are many types of water rock reactions that exist on Earth, and that we are constantly finding new ones on the seafloor as we explore new geological features and regimes. And what I am most interested in as a microbiologist is understanding how those water rock reactions can potentially support life on Earth. So hopefully those videos can help you understand that. Now, I put that question mark after life because, again, I want to emphasize that even in some of these energy-rich environments, other requirements, such as temperature or pH conditions, do not allow life to use that energy. And I think that's a really important thing to keep in mind when we're searching for life beyond Earth. So this is just some beautiful imagery of many of the places on the seafloor that I've had the privilege of studying to remind you they look different because these underlying water rock reactions and the conditions under which they're occurring are different. And so what I want to do for the second half here is really dig into what do I mean when I say energy. So the chemical energy that's being released in these systems, and then how life can use them to make a living. And then we will step back again and try to take that into the solar system before we pass it on to Kevin. So microbes on Earth basically pass electrons between a donor and an acceptor, and that helps them run a reaction that produces energy for growth. And shown here are a few of the chemical reactions that we know microbes here in the deep ocean use. And importantly, you'll note that I don't have light anywhere on these slides. So none of these organisms are photosynthetic in the deep ocean. Instead, they are using chemical energy. And most of these reactions are autotrophic, meaning the organism is fixing their own carbon rather than getting that carbon from another organism. So all of us, we are heterotrophs. Maybe we eat plants, or maybe we eat the cow that ate the plant. And there are many microbes in the ocean that eat heterotrophs, or that are heterotrophs, but most of the microbes we think about in these hydrothermal systems are autotrophic. Now, there's an exception shown here as OM oxidation, organic matter oxidation. I'm going to return to that in just a minute. I did want to mention that when we're thinking about life beyond Earth, though, we might want to consider things beyond oxygen, such as hydrogen peroxide. I'm guessing Kevin will speak to this a bit, but without photosynthesis in these outer moons in particular, we can't get oxygen in the traditional way as we do here. So scientists have been examining the possibility of radiolytic production of oxygen or peroxide into these other ocean worlds. Here on Earth, we focus on oxygen, but there are many places on Earth where there isn't oxygen, particularly at the high temperature end of these hydrothermal systems. And so we also need to look at some of these anaerobic processes, which I've shown here, and include things like methanogenesis, sulfate reduction, methane oxidation anaerobically, and iron reduction. But again, I still have organic matter oxidation here, and I just wanted to briefly mention why I used to make this slide without organic matter, but recently I've decided to bring it back into the equation. As I said, when we think about hydrothermal systems, we traditionally focus on chemolitho autotrophy, but I told you earlier on that we know some of these systems, particularly the ultramafic systems, can produce low molecular weight organic compounds. And we're just beginning to understand in these ultramafic systems how microbes can use those simple organic molecules. Furthermore, a good bit of our work in the ridge flank regions, so away from the mid-ocean ridges, indicates that as seawater circulates through the crust, the carbon, the organic carbon, changes a lot. But it appears that even when it gets really old and really gross, like really chewed down, all the good bits are gone, microbes still seem to like eating it. And so we need to keep this in mind as we think about any ocean cycling through any crust, no matter where we are. And this was really highlighted to me in two recent studies that came out just in the last few months. I was not a part of either of these. Both involved looking at deeply buried rocks, one in really old basalt rock and the other in gabbros, hundreds of meters beneath the seafloor. And in both of these studies, they were able to show that it appears the dominant carbon process going on with these microbial communities is actually heterotrophy. So I want to make sure we keep that in mind regardless of the source of organic carbon on these other ocean worlds. So let's go back into all of these reactions and maybe just kind of step through a potential scenario from another ocean world that we'll hear more about from Kevin. So let's assume we can't find evidence for these oxidants like oxygen or hydrogen peroxide. So now we find ourselves in an anoxic world. Well, microbes have no problem with that, and I'm shown here in green those dominant anaerobic metabolisms that could still occur without oxygen. But then you can imagine through various detection strategies, maybe it turns out that there aren't any of those simple organic compounds, and so we need to return to our autotrophic anaerobic metabolisms. And then let's say we can't find iron, and basically you can step through this. Maybe there's no sulfate, although I know you're going to hear from Kevin about evidence for sulfate predominantly on Europa. One metabolism we're left with, which we hear a lot about in the astrobiological community, rightly so I would argue, is then methanogenesis. So these are organisms that take hydrogen and CO2 and make methane, and these are very successful organisms here on Earth. They're in the human gut, and these hydrogenotrophic methanogens are very abundant at a variety of deep sea settings. And in fact, with the data that Cassini collected from Enceladus, they infer that because of the detection of hydrogen and methane, that hydrogenotrophic methanogenesis might be occurring, however, at a pretty high pH, a pH between about 9 and 11, which is similar to a few, but not many places here on Earth. And I want to make a plug for graduate student Christine Ray. She and her advisors, Chris Glein, Hunter Waite, they have been doing some really neat modeling work, trying to take a lot of the metabolisms that I'm presenting here and making some predictions for a variety of different scenarios on Enceladus and how likely it is that they might be occurring, and if so, how much biomass could be supported. So that work is currently in review, and I would stay tuned for that. But I do also want to remind you that we could be missing metabolisms. You know, as microbiologists, we annotate genomes, and we do a really bad job because there's so many things that we still don't understand the function of. And this is just one example from a paper recently published by Jan Ahmed et al., saying maybe there's this other metabolism out there that, that we haven't detected yet, and it is called sulfur comproportionation, which is quite a mouthful. But in this paper, they show that it is energetically favorable at low temperatures and low pHs, and they suggest looking at places like sulfidic karst systems, shallow marine vents, and other acid-rich, acid-sulfur-rich environments, perhaps to look for evidence of this metabolism occurring. Again, the grand challenge in astrobiology is searching for things, you know, we don't yet know exist. And finally, I focused a lot on energy today, but of course it isn't just about the presence of energy. Cells face many challenges surviving even on Earth's ocean. First and foremost, just because there's energy there does not mean that they can extract it. They have to be able to maintain a disequilibrium. They have to be able to maintain a cell membrane and bring those electron acceptors and donors inside and out of their cell. And we have to think about the flux of those energy sources and other compounds, right? We need to think about how those fluids are moving through the crust, what the rate of reaction might be, what the standing stock might be, what the age of the ocean and the system is, and other things like that. Also, microbes need things besides energy. They also need trace nutrients. They need things like nitrate, phosphate as another great example. Working with Rika Anderson at Carleton College, we have evidence that some microbes in particular vent systems are really starved for phosphate, and in some cases might be turning on and looking for arsenic as a replacement. And of course, the variety of conditions that life can survive in, as I mentioned at the beginning, we have found energy-rich environments without a lot of life, and that's because of these abiotic conditions not being met. Maybe it's too hot, maybe it's too cold, maybe the pH isn't right, but that is, of course, something we have to consider. And finally, I just want to keep it on people's radars to think about viruses because, you know, microbial life on Earth coexists with viruses. And as we think about biosignature detection and looking for life beyond, I think it's really important to keep viruses in mind. You know, through our work over the last 20 years in the deep ocean, we are really pushing the limits for what we know about life here on Earth and beyond. And we have found, we being the royal we, not me, evidence for life 2.5 kilometers beneath the seafloor in 100-million-year-old crust. We see microbes that are thriving in oceanic crust, which was erupted and emplaced only days before. And we're really expanding the known diversity of life on Earth, not just filling in the branches of that tree of life, but also in the types of metabolisms and physiology these organisms have and can survive with. And so I just want to close out with one of the most amazing sights I've ever seen on the seafloor. This is, hopefully you can see bubbling yellow goopy stuff on the seafloor. This is from Daikoku volcano. This is video footage taken by the Schmidt Ocean Institute. And this is over on the Mariana Arc where the Pacific plate is subducting. And when that happens, it remelts and it produces these really gassy, low pH, super interesting volcanoes. And all that yellow and gray stuff you see is actually molten sulfur. This type of volcanism is thought to have been really common early in Earth's history when Earth was young and our volcanoes were hotter. And now it's actually relatively rare, but we have seen it a few times along the Mariana Arc. But I show this video because when we live stream this back to shore, we had about 2 million viewers see this video. And one of the most common questions we got from those viewers was, is this Earth? And really, it's impossible not to look at this footage and draw some parallels to planetary bodies here we have on Earth. And that includes Io, which is the most geologically active object in our solar system, but it isn't driven by plate tectonics. It's driven by tidal friction, as you'll be hearing about from Kevin. And these volcanoes produce plumes of sulfur, that element you just saw in the previous video, which leaves it coated in sulfur. And I think this is just one of the many examples of the parallels we can draw between exploring our oceans here on Earth and beyond. And so with that, I'll say thank you, and I will stop sharing my screen and hand it over to Kevin.
Thanks, Julie. I'd actually like to take a minute here and just pause, in case there are any specific questions for Julie that people want to ask now, but again, noting that we'll have a broader follow-up after Kevin's talk as well. So feel free to, sure.
Hey, Julie, this is Roland. Thanks a lot for the talk. I was wondering, so photosystem 2 is obviously the most efficient way of producing molecular oxygen, but there's at least 5 other ways how molecular oxygen can be produced by life. And so I was wondering if in the deep sea there's any higher concentrations of chloride anywhere, so that chloride dismutase could produce molecular oxygen, and if anything is known about that on other water bodies in the solar system.
So you're asking if there is chloride concentrations higher than what we have in seawater?
I don't think you need particularly high concentration. It's chlorite with a T.
Oh, chlorite.
So ClO2,
okay.
Yeah, ClO2
okay.
minus. Which
I don't know.
I do not know what the concentrations are in the deep sea. So I was just wondering if you would know.
I don't know the concentrations. I do know that you get, phase separation in the deep sea, which really messes with the salt balance and all those things, but chlorite, I don't know. It's a good question.
Okay. Thanks.
No one else is chomping at the bit. I have a really basic question, Julie, and this is somebody far more on the tectonic side of things. Is it possible that there have been other emergences of life on Earth than the current, Earth life we look at today? And, we know that we live on a habitable planet now. Are there any efforts to look for life that is very different than common Earth life on Earth, or is that just a silly thing to do?
No, there's a whole group of people interested in what we just call weird life. Right, life that might not fit into our tree of life. The problem is we don't know how to find it, right? I will say that the tree that I showed could be built on 1,000 different trees, right? And that life is just gone. And what we are seeing now is all that's left, but it has recorded this long evolutionary history. Could there have been many starts and fails and restarts? Sure. What we have now is what we can fit into that singular tree. Viruses present a great challenge because they don't have those conserved genes that fit on a tree, and they mutate very fast. So they are one example where they are very weird life, even though many people don't even consider them life. But certainly scientists have been looking at, synthetic nucleic acids, tweaking the DNA code. Can you replace phosphate with boron or arsenic or things like that? And so I think it's an active area of research, but when we think about going out into the solar system, what do we want to hedge our bets on is really what we have to decide.
That's great, Julie, thanks. We have time for one more pointed question for Julie, if there is one. I'll give 5 seconds for anybody to chime in.
It looks like there's a question in the chat about if, for the Mariana Arc, if there were any heavy metal blebs, such as mercury, along with those sulfur deposits. I don't know the answer to that question. Those little structures that form when the sulfur comes out were actually really hard to collect, but there are some geologists in New Zealand working on that. And, oh, I see, these are going to me privately. That's why you can't see them, Sam. I just figured that out. And then there was also a question about why viruses for astrobiology. And I brought them up simply because in the ocean, for example, if they are orders of magnitude more abundant than microbes, and there's something we can look at under the microscope, and they play a really important role in the evolution of microbes on Earth, and so there's good reason to think that if microbes exist elsewhere, that viruses do also. So with that, I will stop.
Great. Okay, we can squeeze in this last question, and that's it.
Oh.
Were there any life near mid-ocean ridges other than the hydrothermal vents?
Well, I mean, there's lots of life in the deep sea. I think that's what the person is asking. But, you know, definitely hydrothermal vents are hotspots of activity, and you actually see a lot of deep sea life come around to feed off of it because it's sort of like the rainforest in the desert, you might think. But even in the most remote regions of the ocean, far away from any active volcanism, any coastline, you find millions of microbes, you find worms, you find lots of invertebrates, you find viruses, and there, you know, we have not even drilled deep enough to find zero life, so it's everywhere.
Great. Thanks so much, Julie. This is a fantastic presentation, and hopefully you can stick around until after Kevin's talk if there's some cross-cutting questions.
Okay.
And with that, I'd like to introduce Kevin Hand, who's a scientist at NASA's Jet Propulsion Laboratory, and is going to be telling us about the seafloor and oceans of the outer solar system.
Thank you so much. Thank you so much for organizing this. And it's Julie and I have known each other forever, and it's a lot of fun. We've done presentations together, so this, even though virtual, feels like old times. And it's really, really encouraging. I think the last count I saw was like 205 people, which is just fantastic. This is such a great group, a great organization of people that's in its early stages of condensing and coalescing, and we really need to harness a lot of the enthusiasm and energy for ocean worlds moving forward. And to that end, I just wanted to emphasize, this is a slide that I put together years ago on some of the aspects of ocean world exploration. I'm not going to go through these in detail, but it's important to appreciate that at the high level, any framework for solar system exploration has to have a science component to it, coupled with the technology component. The Mars exploration program is a great example of this. And of course, when it comes to the science, we have this aspect of potentially finding an independent origin of life, an extant form of life, where we could study its biochemistry and understand whether or not the DNA, RNA, protein game is the only game in town. But coupled with that, we've got comparative oceanography that will spark new discoveries regardless of habitability. And then, of course, we've got the technology. And my favorite aspect of that is the win-win of exploring Earth as a bridge to exploring ocean moons. Okay, now going from Julie's talk to the solar system, it's important to keep a bit of context here. So from Earth to beyond, some of you might be familiar with this classic book, Consider a Spherical Cow. This is sort of one of those kind of Fermi paradox or Fermi problem books. And on Earth, we of course can go to places easily, relatively easily, collect samples and analyze them in detail. When it comes to planetary science, we don't quite have that luxury. On Mars, we're getting to that kind of in situ fidelity. But with the ocean worlds, we've had the Galileo spacecraft and the Cassini spacecraft and the Huygens lander, and of course the Voyager and Pioneers and Junos out at Jupiter. But we really are quite limited to flyby spacecraft and telescopic observations. So I'm going to today briefly go through, and I apologize, I'm probably going to speak relatively fast. I think Sam said he's recording this, and thus on YouTube, future people can slow it down by half a notch or whatever, but I'll keep the baud rate high so we can get the questions fast. And I threw together a bunch of slides, so I need to make sure I get through these. So there's the agenda, just 3 key things. And more than anything, I want to kind of advertise some of the literature that's out there. The authors that I list below have done a tremendous amount of great work on ocean world seafloors. And that, of course, is a partial list. But in particular, I'll acknowledge Misha Zolotoff and Jeff Cargill's chapter in the Europa book. Quite a good chapter with lots of good references in there. Though many ocean worlds, many candidate ocean worlds have been proposed, these are the ones, as Julie showed, that I like to highlight with, of course, the Earth at the center and then these 6 moons around the Earth. One could add Pluto to this list now. One might be able to add Ceres. There's some interesting developments with the data and modeling of Ceres indicating that perhaps it's got some sort of liquid water region today. If not today, I think the evidence points to a potentially watery past. And coming back to that spherical cow aspect, one of the most useful metrics that one can use to think about seafloors within these ocean worlds is the bulk density. And Hauke Hussmann's paper shown here, or cited here, from 2006, is one of my favorite papers for just kind of laying out the broad context of these many small worlds and moons. And in this plot, Hauke shows the density as a function of radius. And I've kind of partitioned this into a few different regions that help us conceptualize these seafloors. If you're too low in density, like minus, you're too close to the density of water, liquid water or water ice, and you probably don't have enough of a rock fraction to really warrant the definition of a rocky silicate seafloor. So that's in this kind of lower region. Enceladus clocks in at about 1,608 or 9 kilograms per cubic meter. And I'll get into Enceladus in a bit. Pluto in that range of about 2,000. And then when we get up to Europa, we're at about 3,000, and we get perhaps too much rock, too little ice when we think about Io and our moon. Meanwhile, if you get too far out in radius, now you become such a large moon, such a large satellite, that the internal pressures at depth result in icy seafloors potentially. And from a water-rock interaction standpoint, from the standpoint of providing access to the elements and energy needed to build and power life, a lack of a silicate seafloor might be a bad thing. So there's a broad framework for you. Once we factor in a silicate seafloor, we can then start to think about how much energy is being dissipated via tidal dissipation within these worlds. And here I've got Earth, Io, Europa, and Enceladus all shown for context. Earth, here I'm referring just to the seafloor flux. Io, just a stunning world, really a tremendous amount of energy coming out, all resulting from that tidal dissipation. I'm not going to get into what I like to frame as the new Goldilocks of tides versus the traditional habitable zone, but rather I'll just kind of go through relatively quickly some of these high-level parameters that help provide a context for these seafloors. Here are those numbers in a bit more detail: the bulk density, the global heat production, and the average flux. On Europa, and for the rest of the talk, I'm going to focus in on Europa and Enceladus in large part because these are the worlds where we predominantly have, much of the most convincing information, observation, and models that point to silicate rocky seafloors in contact with the liquid water oceans. So at Europa, that average flux ranges between about 10 and possibly as high as 800 milliwatts per square meter. At Enceladus, I've got it listed as 0 to greater than 5,000 milliwatts per square meter. The problem here is that at Enceladus, we've got the south polar terrain where a tremendous amount of power is coming out. And to the north, it doesn't appear to be active at all, at least given the data from Cassini. There might be some modifications to that with ongoing analysis of the Cassini data. But for the most part, the seafloor flux within Enceladus appears to be somewhat heterogeneous, not homogeneous, the way Europa may well be. Partitioning out Europa's energy budget, just to give an example of how to think about tides, within the mantle, and this was a bit of a more conservative upper end number here from 10 to 190 milliwatts. Some fraction goes into the mantle and some fraction goes into the ice shell. This shows that tidal energy dissipation relative to the solar flux, what's reflected, what's absorbed, and the charged particle irradiation, which I won't get into today, but that's really how I spend much of my time, is considering the oxidant production via that charged particle irradiation. And within Europa's mantle, we're talking about 4 to possibly as much as 10 milliwatts per square meter from relic radiogenic decay. Just broadly, when we think about active seafloors and tides, a useful framework is Hooke's law. This is an oversimplification, but where the community now stands is we know that Europa and Enceladus and many of these other ocean worlds have a lot of tidal dissipation occurring within them. But we don't yet have a good understanding of what fraction of that tidal energy is being dissipated in the ice shell versus in a rocky mantle and seafloor. And I like to think of it kind of like Hooke's law, where you've got 2 springs with a mass, and those springs have different spring constants, and the spring constants determine how much heating occurs within those springs. So in this analogy, the stretching and pulling that worlds like Europa and Enceladus feel from Jupiter and the moons with which they're in residence gives rise to stretching and pulling of the ice shell and mantle, where we're still trying to figure out the spring constants and how much dissipation occurs within those 2 regions. Now, in a sort of ideal scenario for habitability, we've got some partitioning of tidal dissipation in the ice and some in the rocky interior, which could potentially give rise to a cross-section like this, where you've got ice cycling with the ocean, fracturing, allowing oceanic material to come up to the surface, and then active hydrothermalism on a rocky seafloor. So both regions get part of that tidal energy. From an energy dynamic standpoint, you can envision 3 different kind of scenarios. One in which the ice shell receives most of the tidal energy. And in that case, you would likely have an oxidized ocean because most of the cycling occurs between the ocean and the ice, bringing in some of those oxidants that are radiolytically produced on the surface. Meanwhile, if most of the tidal energy is partitioned into the seafloor and mantle, then you may well have a reduced ocean, an ocean dominated by reductants coming out of hydrothermal vents and active regions on the seafloor. And if we do indeed have a sustained, habitable, biological ocean, then perhaps part of the seafloor is hydrothermally active and part of the ice shell is active, allowing oxidants from above and reductants from below to maintain a chemically rich ocean that has the kind of chemical disequilibrium that Julie spoke to. Okay, so getting into some of the dynamics of what we actually think we know about both Enceladus and Europa, really it's going to boil down to what we have learned about salts and the fingerprint that salts provide for us of potentially active oceans cycling with seafloors below. And shown here is a table from Jeff Cargill's classic paper where he looked at all sorts of different compositions and leaching and how you just kind of take chondrites and make a moon. And here he was considering Europa specifically. There's been a lot of great work since this time, but Jeff really kind of helped set the stage with this. What you see is that when it comes to just making a moon out of chondrites, space rocks, if you leach them all together, you'll end up with quite an abundance of magnesium and sulfate. Meanwhile, within Earth's seawater, we know that Earth's ocean is not dominated by magnesium. It's dominated by sodium and potassium. And in addition to that, Earth's seawater is not dominated by sulfate. It's dominated by chloride. And so there's this kind of difference between a primordial condition that might arise for an ocean versus what an ocean eventually looks like from a salt composition standpoint after it's cycled through with a rocky silicate seafloor. In the interest of time, I'm not going to go through this in detail, but what you're seeing here is the efficiency of the drawdown of magnesium at various temperatures and different pressures. And this is a model that I made, I think, using Supercrit many years ago. And then I looked at the time scale for magnesium sulfate drawdown through Europa's seafloor for a variety of fluxes, a variety of cycling. And basically what this plot on the right is telling you is that even in very, very low flow of oceanic water through Europa's seafloor, you still draw down magnesium sulfate very efficiently. And really what we're getting at here is that a cation like magnesium and an anion like sulfate get drawn out of seawater, whereas sodium and chloride have very long residence times. And so to zeroth order, magnesium sulfate is a bit of a harbinger of a stagnant primordial ocean, whereas sodium chloride is, again, to zeroth order. This is a much more complex story than the way I'm framing it in the interest of time, but sodium chloride is a bit of a harbinger of an ocean that is active today and cycling with the seafloor. So what do we think we know about Enceladus? Pardon me, wet my whistle a bit here. And I apologize if you're hearing noise from the background. They started construction on the neighboring house just last week. Hopefully this directional mic is zeroing that out. So at Enceladus, the Cassini team, and the INMS team, and the CDA team on Cassini have just led to a tremendous amount of information that have helped kind of transform Enceladus from that spherical cow to one in which we kind of now think we actually have some really good observations of the composition of the plumes and the E-rings, and then by association, hopefully, the ocean within. And very briefly, I'm going to go through each of these plots in kind of a time succession. So when Hunter and team first released the INMS data, we were all tremendously excited. I was not on the Cassini team, but my jaw still dropped. They found short chain organics and lots of interesting volatiles. But being a skeptic, I and others were kind of like, well, that's a bit comet-y. It's not necessarily indicative of an ocean below, and in particular an ocean cycling with a rocky seafloor. Fast forward to some of the CDA results, and now we've got evidence for salts, and not just any kind of salts, but sodium and chloride salts. And so with the cosmic dust analyzer, now folks like myself and a few other skeptics on the initial data from INMS began to get convinced that we do have evidence of a salty ocean and possibly even an ocean that's interacting with an active seafloor. And then following on that, we have, of course, the publication of the silicon nanograins, which is another indication of water-rock interaction, and even of mild, roughly 90 C or so hydrothermal alteration and interactions occurring within a silicate seafloor. And then most recently, the very meticulous, I'm still a little bit skeptical about this, it was an incredibly detailed and hard study to pull off, but the observation of excess hydrogen in the plume region around Enceladus really is quite an exciting result about ongoing possible hydrothermalism within Enceladus. Might there be other explanations for this concatenation of evidence? Yes, but at this point, even myself trying to look for ways to explain away the evidence, you know, it's really kind of a special pleading case, in my opinion, to argue against Enceladus having a salty liquid water ocean in contact with at least a mildly hydrothermally active rocky seafloor. And there's all sorts of great modeling work that goes along with this. Here I'm focusing predominantly on the observations. Okay, what about Europa? Here's Europa. The first thing to appreciate, for those of you who are not coming into the Network for Ocean Worlds from a planetary science background, is that Europa has very few craters, and unlike Enceladus, Europa has a really odd diversity of geologic structures, and I think Cynthia Phillips spoke to this in one of the earlier presentations given to now. I'm going to focus in on some of the discoloration that is seen within Europa's ice. Curiously, we do not see this kind of discoloration out at Enceladus, and there may be a few different reasons why. So what is that composition? What is the composition of the dark material? Some of it we do have good evidence is exogenous sulfur coming from those volcanoes on Io. I won't get into detail here on that. Some of it we do think is actually magnesium sulfate, but those second two bullet points speak to some of the debate about magnesium sulfate. In partnership with Mike Brown, published a paper back in 2013 where we actually argue that the magnesium sulfate that we think we're seeing spectroscopically on the trailing hemisphere of Europa results from endogenous magnesium coming from the ocean, coupled with some exogenous sulfur radiolytically processed into magnesium sulfate hydrate. I'm not going to talk about that today. Rather, I want to focus in on something else altogether. What is that reddish yellow-brown material? Well, that takes us to the lab, to our Ocean World lab at JPL. You can go to the website here to kind of get a better sense of our Europa or Enceladus in a can. We can't wake up in the morning and go to these worlds, so we got to recreate them in the lab, which sadly I haven't seen in many months. Okay, so here's a bit of simulated Europa water with sodium chloride. This is ultra pure deionized water with reagent grade sodium chloride dissolved in it. I'm pumping out the water, and this is within one of those chambers bubbling away. This curiously might be what some of the surface of Europa looks like, or Enceladus, within some of the fractures where bubbling is happening. After all that water was evolved away, I was left with this evaporite, which then I irradiated with an electron gun, similar to what occurs on the surface of Europa. And sodium chloride is kind of frustrating for spectroscopists. It's just a flat spectrum. It looks white, as you know from looking at salt on the dinner room table. But after irradiation, that salt traps alkali halides like sodium chloride, traps electrons in various color centers, yielding a yellowish, brownish, reddish color to that irradiated sodium chloride. And I like to think of this as kind of an invisible ink experiment where the surface of Europa and the surface of Enceladus, for that matter, has sodium chloride mixed into the surface ice. But then with the irradiation, you can see that sodium chloride, whereas you cannot spectroscopically see it prior to irradiation. And just last summer, Samantha Trumbo, a brilliant grad student at Caltech, working with Mike Brown and myself, she managed to get time on the Hubble Space Telescope. This isn't last summer, this was a while ago, but the publication came out last summer. And Samantha actually found in the Hubble Space Telescope data this absorption feature. So the black lines here, that's Hubble data, and the red line here is one of my lab spectra. And this absorption is highly indicative of that irradiated sodium chloride. And so at Europa, this observation from Hubble, I think, supersedes all previous observations from Galileo or ground-based observations, and serves as our best evidence today of Europa having a liquid water ocean that is interacting with a rocky silicate seafloor. Now, just as a little asterisk to that, you might say, well, what about the magnetometer data? Well, the magnetometer data, of course, indicates that there's a conducting near-surface layer, but the magnetometer data is a little bit immune to whether or not that salt is magnesium sulfate or sodium chloride. Here, with this specific absorption feature, I think we've got really strong evidence for sodium chloride, which again, I think argues to an active ocean cycling with a seafloor. Okay, I'll just finish up with a quick highlight of some stuff about our own ocean. This just gives you a sense of how the pressures within Europa and Enceladus compare to the depths of our ocean. The gravity at Enceladus is so low, less than 1% of Earth's, that going a kilometer down in our ocean is equivalent to getting deep within Enceladus. Meanwhile, at Europa, if you do the math, it turns out that about 11 kilometers down in our ocean is roughly equivalent to about 80 kilometers down beneath the surface of Europa. And that 11-kilometer depth is the Mariana Trench. So I'll finish just showing you some highlights from an expedition in 2012 that I was a science team member of. And we deployed these landers built by Kevin Hardy, shown there at bottom left. And we managed to get a view of the deepest region of our own ocean. And this little video, hopefully it's playing. I'll wait just a couple of seconds because I think there's a bit of a lag. That video gives you a view into the Sirena Deep, a region within the Mariana Trench, where we serendipitously landed and found outcrop and talus blocks. And when we zoomed in on those talus blocks, we saw these very bizarre filamentous formations coming off of these blocks. And I'll just end by referring you to this paper. It just came out in April. And in that paper, we go into some of the evidence that we have for this region undergoing serpentinization and these filaments possibly being microbial in nature. And coming back to one of the questions that was asked earlier about the seafloor, a lot of the open ocean and what is surviving in the abyssal regions in the hadal depths of our ocean may well be fed by marine snow. In other words, stuff coming in from above. But here, if indeed these filaments are microbial mats, we think that this is evidence for some of the deepest, highest pressure chemolithoautotrophic microbial activity going on within our own ocean. And some of the amphipods that we observed, we think, then feed on some of these filaments and then provide, these filaments then provide that base of the food chain. So I'll finish there, and thanks so much, Sam. Hopefully we've got time for questions. Good, we're at 20, good.
Absolutely, Kevin. Yeah, that was a really wonderful talk. Thanks for the summary and also, as always, the fascinating imagery and movies. I'd like to start off, if there are specific questions for Kevin, we'll start with those. And if there aren't, we can have a broader discussion about questions that might be pertinent to both Kevin and Julie.
I have a question.
Go for it.
Yeah, hey, Kevin, nice to see you, and that was a really great talk. Really like the point about magnesium sulfate being cycled into the seafloor, and thereby sodium chloride being an indicator of an active seafloor. But I wonder about a similar process happening in the ice, and that is the preferential incorporation of sodium chloride into the ice. The fact that sodium chloride is much lower melting temperature than magnesium sulfate might suggest that as Europa's ice freezes, it preferentially incorporates the sodium chloride. We made this point in a paper in Astrobiology in 2019.
Hey, Steve, can you hear me? I just unmuted. Yeah, can you just repeat the last? It cut out there for a second. So the incorporation of the sodium chloride in the ice, but not the magnesium sulfate.
Yeah, and so yeah, I mean, this is something that I've been wondering about for a while, and I did some freeze-chem calculations similar to what Marianne did in 2005. Yeah.
Okay, I got your question now. Yeah, in terms of the ion exclusion, So, so what Steve's referring to here is at the base of the ice shell. If you've got, say, slow basal freezing, depending on the freeze rate, you can get inclusion or exclusion of different salts. Divalent versus monovalent may have preferential inclusion or exclusion. And like you're saying, Steve, it appears that monovalent salts like sodium chloride could get preferentially incorporated, which then might bias our interpretation of the surface fingerprint, right? Steve, that's where you're going with that? Sorry to ask you to unmute again. Hey. Oh, you're still on mute.
Sorry, confusing the camera and then the mute. Yes, that's where I'm going with that.
Yeah, and so you're spot on. And just to kind of highlight a little bit of that magnesium sulfate story, coming back to that 2013 paper, a part of what we developed there is kind of a mixed magnesium sulfur cycle. And so absolutely the surface expression is a biased lens through which to then infer the ocean composition. Now, if plumes are the way that everything gets up and out, then that's a whole different partitioning as opposed to basal freezing. So, I will plead the spherical cow on this. We just got to get back out there.
Great, thanks a lot.
Thanks.
Thanks, Steve. Real quick, I'm happy people are staying for the discussions, and again wanted to thank everybody for joining us today, both inside the network and outside. And just in case people have to tune off, I want to mention that our next quarterly lecture series will be on the ice and ice-ocean interface of Earth and planetary bodies. And just again, remind steering committee members real quickly to please check your email and respond about the decadal white paper, because so much of what we can do together is advocate for better exploration of Earth's oceans and our ocean worlds. With that, happy to go back to taking questions for Kevin, or if there are questions for Kevin, Julie, or both about the cross-cutting nature of studying ocean worlds, that's great too. We have one question in the chat on what yields more accurate results, analog sites of icy moons or computer simulations.
It's just it's a state machine of they have to feed each other, and then every now and then we get to insert some real spacecraft data. So if we're lucky, we get a mission once a decade. Hopefully, we can start to improve on that pace, but. I think the analog studies, certainly when it comes to astrobiology, are critical. There's the old adage in geology that the person who's seen the most rocks is the best geologist. Now, you can debate that, I like to adopt that for astrobiology, which is that, you know, the person who's seen the most microbes is the best microbiologist or astrobiologist. Again, lots of caveats there, but I think it does really help set the stage to really examine these environments that Julie spent a lot of time detailing. Julie, what do you think about that question?
Yeah, I think that's a really tough question, and I'm always amazed that stuff I look at on the seafloor, I make some assumptions about it, and then I work with stuff in my lab, the materials, and I was wrong. And I think it goes to these challenges of doing all these remote observations. You know, ground truthing without samples in hand is really hard. And one thing I cut from my talk in the interest of time is that as oceanographers, we're really lucky because we mostly get to go down there and bring samples back. But we also recognize that isn't always possible, and we'd get a lot more data if we could do it in situ. And so we've been working towards trying to develop instrumentation to do more in situ experimentation, analysis, and monitoring, which is what you guys always have to do. And, you know, I think trying to bring more of that to the seafloor will only help us when we get to Europa or Enceladus in interpreting the results.
Great. So that kind of ties into Riley's question. In what kinds of experiments shed the most light on process happening on the seafloors of Earth and other ocean worlds? And I guess there's an interesting question there, right? Kevin and Julie, do your labs look the same? I mean, what kinds of experiments are you doing, and what's been the most helpful?
Well, Julie, maybe I'll prompt you, Julie, to maybe share your thoughts on genetic analyses, you know, doing PCR and really building out the tree of life, filtering microbes, and you know, how obviously when we think about searching for life on these other ocean worlds, we cannot a priori assume that a PCR primer is going to find DNA on these worlds. If anything, we're hoping that maybe we find some other informational molecule, some other framework. When it comes to your lab and other biology labs, Julie, how do you use both culturing and genetic techniques to get at the big picture questions that you and others are asking?
Yeah, you know, I've been in Kevin's lab. Our labs don't look anything alike. Mine has to be crazy clean because we are dealing with super low biomass environments often, but it really looks like a molecular biology or a microbiology lab. And Kevin's right, we try to culture these organisms, but that has a lot of bias because you're really only studying what you can grow. And so the person who asked the question of what kind of experiments shed the most light, you know, there's a lot of us doing experiments with kind of model Earth organisms and pushing them into Europa-like or Enceladus-like conditions to see how they cope. But, you know, that's a little bit simplistic, right? Microbes don't live in isolation, first of all, and an Earth microbe might never have seen conditions like what exists on these other ocean worlds. But it's an important kind of step to help us understand how to parameterize sort of the habitability of these microbes. I think the most important thing we do, though, is we go to these places on Earth, and I don't go by myself, right? I go with geologists, geochemists, hydrogeologists, modelers. Sometimes Kevin comes, you know, and we're really trying to bring together a diverse group of minds to figure out how that system works. And I think that is really shedding the most light on how things work here on Earth.
Great. And very interesting answers. So Chris wants to know if chondrites show high concentrations of complex organics, does that have any implications for the potential for heterotrophy of the kind that you were discussing earlier, Julie?
Yeah, well, I don't really know. Kevin can answer the questions if we think chondritic organics would be delivered to these moons, first of all. But I've definitely had to reframe how I think about heterotrophy and hydrothermal systems. And I do think that if you have delivery of external organics, that could be very helpful, but then you have to look at the flux, right? Is it a one-time deal? Is it constantly happening? And things like that. But all of our data indicates, and again, this is the royal hour, both in my lab and other labs, that even microbes that have the ability to fix carbon, a lot of them still carry the genes so that if a piece of organic material comes along, they can chew it up. It's just really delicious and makes life a lot easier.
Great. Are there any other questions for Kevin and or Julie, or both?
Kevin wrote a book, you guys, so if you're into it, he's got this book. It's called Alien Worlds. It's out and about. Go read it.
Alien Oceans. Thanks, Julie.
Alien Oceans, sorry. Sitting on my bedside table.
Great. Well, I'd like to thank you both again so much for being here. You know, it's, I think, a great platform to start reaching out to more of the community. And so thanks again to all the people from inside and outside the network who joined today. Again, next time we'll be talking about life in the ice and ice-ocean interface of Earth and planetary oceans. And also, this isn't a one-person show, so I'd like to thank the now co-leads, Chris German, Alyssa Roden, Allison Murray, and Kevin Arrigo for running the network and allowing this meeting to take place. And also the co-organizers, Ella O'Brien, Shannon MacKenzie, and Laurie Barge, who helped put this lecture series together. So thanks again one more time, Julie and Kevin, and to everybody who joined us today.
Thanks, Sam. Thanks a lot there.